In this video, we're going to be discussing G-protein coupled receptors. And before we talk about the pathways of these receptors, we're going to talk a little bit about the structure. So G-protein coupled receptors are embedded in the cell's phospholipid bilayer. GPCRs are made up of alpha helixes and transverse across the cell membrane 7 times. Because GPCRs have such a complex embedding in the cell membrane, GPCRs are also referred to as 7 transmembrane receptors. The reason why we call them G-protein coupled receptors is because each receptor has a G-protein attached to it, or coupled. The function of G-proteins in GPCRs is to act as a messenger to further relay information. The active form of the G-protein has GTP attached to it, or guanosine triphosphate. The inactive form of G-protein has GDP attached to it. Keep in mind that the G-protein is made up of 3 different subunits: the alpha subunit, beta subunit, and gamma subunit. When the G-protein is activated, the alpha subunit dissociates from the beta and gamma subunits. So now we will discuss the pathways in which G-protein coupled receptors relay information within a cell. First, a water-soluble extracellular chemical messenger or hormone will activate the receptor. The receptor is activated when the hormone acts as a first messenger and binds to the receptor. The receptor has a specific shape to fit the hormone. The binding of this hormone to the receptor will cause the next step in the cascade of events. Once binded, the GPCR will undergo a conformational change and allow for GDP to be replaced with GTP on the G-protein. This causes the G-protein to be in its active form. When the G-protein is activated, the alpha subunit dissociates from the beta and gamma subunits. Once the G-protein is activated, it will target another protein or enzyme and regulate its function, typically by activating it. The purpose of activating this enzyme is to produce an intracellular second messenger, such as cyclic AMP or IP3 and DAG. The type of second messenger will be determined by which form of the alpha subunit is present in the G-protein. The alpha S subunit will produce cyclic AMP, and the alpha Q subunit will give rise to the second messengers IP3 and DAG. The second messenger will tell the cell to carry out specific functions or will trigger a specific cellular response. Today we will be focusing on the alpha subunit as an example, and we will be using the first messenger or chemical messenger, epinephrine, to explain the GPCR signaling mechanism. Epinephrine is going to bind to our G-protein coupled receptor, acting as a first messenger. Once epinephrine binds to the receptor, the GDP that is adhered to the G-protein will be replaced with GTP. From there, the G-protein alpha subunit will dissociate from the gamma and beta subunits. With the replacement from GDP to GTP, the G-protein will be in its activated form. The G-protein will stimulate a membrane protein called adenylate cyclase. When adenylate cyclase is activated, it will convert ATP to cyclic AMP, which will act as our second messenger. Cyclic AMP will activate protein kinase, which will cause a cellular response, such as an increase in heart rate or maybe dilated blood vessels. So in this case, epinephrine was the chemical messenger or first messenger. The GPCR was our receptor. The G protein subunit activated our enzyme, adenylate cyclase, and our second messenger was cyclic AMP. This is just one of many GPCR pathways in the body, and although different, each GPCR pathway follows the same mechanism illustrated in the video, with different chemical messengers or target enzymes.